Hello again, everybody. Welcome to today's CAR-T webinar. I am Mingjuan, the Director of Marketing at uBriGen. We are offering a series of webinars on the basics of advanced therapy, the development and manufacturing. So today is our first webinar, and the topic is CAR-T 101. We are very happy that you can join us today. So you can see on the slide before starting, we have a few logistics. So you will be put on mute during the webinar, but we do welcome you to ask questions using the Q&A box on the top right corner of the platform. So then we will have a Q&A session after the presentation to answer your questions. So it is my honor to introduce today's speaker, Dr. Ken Warrington. Ken is our Executive Director of Strategic Business Development and Head of Technical Services. Ken has extensive expertise in the advanced therapy field. He has a very broad and deep knowledge in many modalities, including live attenuated viruses, virus-like particles, viral vectors, and gene-modified cell therapies. Ken has worked in both academic and industry setting. He served on the faculty at the University of Florida Pediatric Division of Cellular and Molecular Therapy. He then took on numerous leadership roles in the cell and gene therapy industry, including CDMOs, regenerative medicine, and gene therapy businesses. Now it's my pleasure to turn over to Ken. Ken, please take it over. Thank you.
Great. Thanks, Mingjun. And I appreciate everyone's attendance here today. Initiating our series of webinars from an educational as well as a manufacturing insight perspective. The goal is really for this first webinar, is to talk at a high level around the general characteristics of CAR-T therapies, initial manufacturing considerations that we see when we work with early stage companies, and then wrapping up with just general QC and regulatory guidance associated with the development of these therapies. CAR-T cell therapies are obviously making a large impact in the treatment of cancers, and they're generally produced by the isolation and enrichment of a patient's T cells, the activation of those cells to become cytotoxic for the cancer cell, which is targeted through the genetic modification of the T cell to express a chimeric antigen receptor. These cells are then expanded once activated with the goal of ultimately formulating for a reinfusion into the patient. I think to date, we're at 6 approved CAR-T therapies, and these CARs have focused primarily on CD19 or BCMA antigens, and their approvals are based on second or third line therapies for a list of relapsed or refractory leukemias, lymphomas, and in the case of BCMA, multiple myeloma. An initial and important consideration is the development of your CAR. And we've seen over the past few decades important advancements in the generation of novel CARs that have improved affinity for the target antigen, stimulatory and proliferative moieties that induce signal cascades that lead to more robust CAR-T therapies. And the design of the CAR is also of certainly a specific concern from the viewpoint of the regulators. Initial generations lacked the ability to really have robust activation and proliferation. And so what is developed over the years is the improvement of the extracellular domains for targeting antigen recognition in a tunable fashion. The role of the hinge and transmembrane domains as they link the extracellular CAR to the signal cascades involved in epitope recognition, activation strength, have impact on CAR expression, and also T cell cytokine release. An additional and next generation focus has been on the intracellular domains, the addition of more co-stimulatory and activation moieties within the intracellular motifs of the CAR. Target, and then certainly in next generation, we're now targeted production of specific cytokines that aid in the persistence and activation, proliferation of the CAR Ts through increased signaling involving, in particular with the next generation design, very specific cascades that are involved in the CAR T development. Certainly going forward, we're now beginning to see additional focus on the extracellular domains, the ability to potentially target multiple antigens simultaneously, as well as additional arming of the CAR design to express very specific additional proteins. Another area of very active investigation is the delivery approaches used to express your CAR within the target T cell populations. Clearly, the lead has been in the retro and lentiviruses, primarily based on their stable gene expression, the ability to infect target T cells at relatively high efficiencies, and of course, the integration at high efficiencies. Certainly in red, I have some of the downsides of each approach. Viral packaging can be complex, time-consuming, and sometimes expensive. There are packaging limitations depending on the design of the viral vector being used. And of course, with recent warnings as well now, that random insertional mutagenesis is certainly a concern and a consideration in the deployment of viral vectors for the development of CAR T. Additional approaches, active areas of investigation, involve transposons, which have high expression efficiency, have the ability to express multiple genes, but are still affected by low transduction efficiencies and random gene integration. Gene editing approaches are also very actively being investigated, including zinc fingers, TALENs, and CRISPR platforms that lack the insertional mutagenesis, are highly specific in their ability to edit, but tend to also suffer from low editing efficiencies and in some cases high cytotoxicities. The transient expression of plasmid using plasmids and RNA also being investigated, primarily due to their lack of insertional mutagenesis. But again, these tend to suffer from low transduction efficiencies, as well as the persistence of the expression of your CAR due to the transient nature of these approaches. As a manufacturer, this is our general, I think in general, but also our general manufacturing workflow at our Center of Excellence for Cell Therapy in Worcester, Massachusetts. The general manufacturing steps are fairly well established across the field, and our experience within our Worcester facility, led by Joe Sinclair and his technical and analytical teams, have experience with this process established with a track record of IND approvals. Initially, the leukapheresis product is received. the next steps in the production in these workflows involve T cell enrichment, followed by the activation of these T cells prior to CAR transduction, regardless of your approach to CAR delivery. These cells are then washed, expanded, harvested, and final formulation, and if required, a cryopreservation in order to then ship to the patient's treatment center for reinfusion into the patient. And what we see in general as we work with early stage companies and our general manufacturing considerations is that often we're looking at a transfer of an academic or research grade process at small scale. And so we focus initially on optimization of protocols to incorporate better process closure, the use of prodigies and Cocoon, et cetera, to close the process, at least at T cell enrichment, activation, and transduction. And we really focus in evaluating the needs of any program on some very specific key data points in this evaluation. Given the variability in donor T cell populations and the various variability also in the disease burden that the patient may be experiencing, understanding the populations in the starting material, then ensuring that our cell numbers are sufficient at transduction, and evaluation of the transduction efficiency, regardless of the platform being used to deliver, and then ultimately the percent of CAR T positive cells and the time it takes to expand the cells to achieve the required dose to support the early stage clinical trials. This is a general summary of considerations with respect to QC release panels for CAR T. Obviously, rapid methods are required given the turnaround. And then a general summary of the regulatory guidance that was recently issued by the FDA. Typically, you're always looking at safety, impurities when it comes to the release panel for a CAR T product. In addition, cell viability, the percent of specific T cells and their ratio to actually those T cells that are expressing the CAR, obviously an important consideration. And then in particular with the use of viral vectors, vector copy number, the potential for a replication competent vector in the preparation, and then obviously one of the more important and more challenging requirements for CAR T therapies is potency, as these can be very challenging assays to develop, focusing on the cytotoxicity of the CAR T towards the target cell type and the cytokine production that is being released during the treatment of these target cells. The FDA's guidance has really focused in on some very key areas, in particular the CAR construct, an assessment of the antigen recognition domain and its specificity to the target antigen, an assessment of the functionality of the various signaling domains, and then, of course, the impact of any additional domains on the CAR's specificity and activity are areas of focus in terms of design of your CAR construct and assessment of its functionality. The vector, of course, what type, structure, as well as a justification for any additional functional elements that are incorporated other than the CAR itself. Of course, using various transduction platforms, there's considerations around the manufacturing, release, and stability testing of these delivery methods, and any key raw materials that are required to support the production of your vector, including the manufacturing and characterization of cell banks used in their production. Plasmid DNAs that are used in their production are also a focus of the guidance. Cellular starting material is a challenging one, and of course, the FDA would like to see a risk assessment of any previously administered CAR T product. Some of these patients have relapsed following an initial treatment, and so the evaluation of residual CAR T and vector copy number for both the new and any previously administered CAR Ts in both the starting and final material is an area of focus. And then specific acceptance criteria, including minimum cell numbers, the viability, and the percent target T cells. The CAR T final product is really focused on, the guidance focuses on manufacturing in process controls. Having an optimized manufacturing process engineered to maintain both integrity and potency of the therapy, rigorous controls implemented with stringently throughout the manufacturing process to ensure both quality and potency. Very stringent material control in the manufacturing process to address any issues that may affect reproducibility as well as potency. And then again, a reliable potency lot release assay that requires the utilization of advanced techniques to test product lots for consistency. uBriGene has a track record of producing CAR Ts and a variety of the vectors required to transduce T cells in the development of CAR T therapies. Our Center of Excellence in Worcester, Massachusetts, is about a 27,000 square foot facility, again led by Joe Sinclair. There are 4 commercial-ready GMP clean rooms to support both your process development, preclinical, and clinical trials for all cell therapy programs. While we have extensive experience with CAR T, we also have the capabilities and are establishing new production workflows for a variety of cell therapies. And we offer comprehensive QA and QC and analytical capabilities to allow full scope of testing for your final products. Our plasmids and viral vectors are produced in 2 China facilities in Jinan and Suzhou with large-scale capabilities in fermentation and mammalian cell culture with numerous vector production lines to seamlessly transfer manufactured CAR delivery platforms to the Worcester site for additional manufacturing of the CAR T. Our platforms are broad and covering most of the advanced therapy medicinal products in development, from plasmids and vectors, various cell therapy products, as I mentioned, all the QC release and process development, process and assay development, and more recently RNA products, including mRNA as well as circular RNA. And we have developed a LNP to assist in the delivery of these products. Just a little deeper dive, illustrating some of these product highlights that really focus in on CAR T. We have the capabilities to produce clinical grade GMP plasmid and also offer clients access to lentiviral helper plasmids off the shelf of GMP quality with drug master files on file with the FDA. Our extensive experience in the production of lentivirus, also offering access to a 293 cell line to assist with the identification and procurement of key raw materials, both cell lines and plasmids to produce your lenti. This is all part of our ongoing development that we're also have worked extensively in cryopreservation solution formulation to improve overall lentiviral stability. As I mentioned, in our Center of Excellence in Worcester, while we have a more extensive track record with CAR T, We have the capabilities to manufacture any type of CAR product, CAR NK, CAR macrophage, etc., and have initiated programs and workflows around iPSC development. And as I mentioned, more recently, we now have capabilities to produce RNA, and look forward to advancing those technologies and offerings to our clients. So with that, I'll wrap up, provide contact information here. If you'd like to reach out to myself, listed there, or our general contact at uBriGene, the email and phone number are also listed. And once again, I appreciate your attention and attendance for this initial introduction to CAR-T and uBriGene's capabilities. And I'd be happy to field any questions. Thanks.
Thank you so much, Ken, for a very informative presentation. Yeah, there are a few questions coming up. CAR-T manufacturing is a complex process, and the CAR-T assays, quality control assays, are critical, and there are a few questions regarding it. The first question is, so there are, among all the CAR-T release assays, what assays will be conducted on-site at your facility, and what assays will be outsourced? Ken.
Yeah, good question. We offer the full QC release, analytical development capabilities within our own facilities. In particular, the more standard identity, safety, and purity profiling are well established. And I think important to mention that our analytical development team in Worcester has extensive experience, and we have in-house capabilities for the development of potency assays. And this is where we're fielding a lot of requests. And we have really assembled a great team and a full suite of instrumentation to allow us to tackle all those methods that require evaluation of cytotoxicity using flow-based assays, as well as the expression of very specific cytokines involved in the potency of these therapies.
Thank you, King. I think you just answered the question. The second question regarding the potency assay, the killing potency assay. So the question is, what type of the CAR T cell killing assay we can perform, and using what equipment? I think you just answered, is that right, Ken?
Yeah, I mean, we're certainly eager to and open to tackling any request when it comes to cell-based potency assays. But yeah, in general, these are, tend to be co-cultured assays involving the CAR T, the target cell expressing the specific antigen. And so they end up being complicated from a culturing standpoint, but using a combination of flow as well as measurements of multiplexing, cytokine release, et cetera. We are open and have the capabilities to perform assay development in that space.
Thank you so much, Ken. There is another question regarding the CAR delivery format, because there are so many options, right? Viral and the non-viral you mentioned in the presentation. There are some advantages and disadvantages. So this customer has a question that they have a typical CAR T project. What does uBriGene recommend the delivery for format, using a viral or RNA? It's becoming popular. So which platform do we recommend to the clients?
Yeah, it's a good question. I think certainly we are agnostic when it comes to the platform. We're happy to and have the capabilities to manufacture a wide variety of these platforms. But if you look, specifically at what's been approved, they've primarily been third generation lentiviruses. I think the main reason, of course, is the stable expression and persistence. And so I think in general, certainly, an offering that we have that involves the ability to supply both qualified cell lines and plasmid for the production of the lenti. But I think, that's an area of active investigation for the other platforms. We're not there yet, but I think, given all the intense effort here, that we will get there. But right now, lentivirus seems to be the lead request, and it has certainly been a part of the approved therapies.
Thank you, Ken. That's very helpful. If you the audience has questions, please type in the Q&A box. This is on the top right corner. Okay, I don't see any more questions. But before closing, if you have any questions and Ken has the last slide or any needs, please send us email. That will be contact@uBriGene.com, or give us a call. The phone number is on the website. And before closing, I would like to conduct a poll. It's one question. Go ahead.
I'm sorry, just to interrupt. I do see a couple of additional questions in the chat side. I'm not sure if you see them, but I'll go ahead and tackle the last two I see here. So one is asking, they would like to know if the manufacturing process of T cells, there's an inactivation step of T cells required to stop the proliferation of T cells after expansion. I think the short answer is no. But this is one of the challenges of these therapies and the importance of having a well-designed CAR, both from a specificity as well as an affinity standpoint. T cells in general persist, I think, approximately a year. But what can occur in the treatment and where you're now seeing development of potentially kill switches and the ability to self-inactivate or somehow turn this off at some point is the side effects that can occur with these therapies. Cytokine release syndrome, neurological effects. And so there isn't generally an inactivation step in manufacturing, but it's certainly an area of investigation on the clinical side in terms of if this is too strong, how do we dim or switch it back down? And so it's a good question, but there's no general inactivation step during manufacturing. And then there's one additional question asking about retrovirus platform. We do offer retroviral manufacturing. It is not as well developed in terms of the ability to offer all the plasmids and off-the-shelf access, et cetera. But we do have experience and capabilities to produce retroviruses as well. And one other question just came up, but I'll wrap it up there. Is your CAR T service or product based on individual CAR T or general? Yeah, it's a good question. Obviously, what I presented is based on autologous because we're not quite there yet with allogeneic, but we will be. And certainly the ability to have universal CAR Ts is an area of active investigation. And certainly would make the logistics of producing and treating patients much easier. But right now, these are autologous platforms that are what we're offering, and so we look forward to working with any future clients that have an allogeneic CAR T that's ready for prime time.
Okay, there's another question regarding RNA. If we had any RNA clients and experience with the GMP manufacturing.
Yes, we do. And we're It's an area of active development for us to make these platform offerings more robust and more plug and play. But yes, we are currently developing RNA, GMP RNA manufacturing. I think initially to focus on the development of iPSCs and the use of RNA for their initial development. And so the short answer is yes, both from mRNA as well as circular RNA.
Okay. Any more questions from the audience? If not, I would like to launch a one question poll. I would be great, appreciate if you could participate. It's just one question. Yeah, if it's just what type of therapeutic programs are you working with? Just click any that you're working with. Just a short question. Okay, I will close the poll. Thank you. As I mentioned earlier, that we have a series of advanced therapy webinars on a monthly basis. Please watch out for your inbox for the next webinar information. Again, thank you, Ken, for a great informative webinar, and thank you, the audience, for joining us today. This will conclude our webinar today, and have a great rest of your day.